Hello everyone, in this video we will be solving the GATE 2024 question paper Food Technology section. Welcome to Udemy. Now to start the video, the very first question is, which one of the following fungi produces aflatoxins? Now this question is from the food microbiology section and the topic is toxins from microbes. Now if we look and if we try to recall that aflatoxins are nothing but actually the type of mycotoxins, and mycotoxins are actually fungal type toxins. So fungal toxins or mycotoxins are of different types. For example, aflatoxins, patulin, ochratoxin, penicillic acid, and various other mycotoxins. And among the different mycotoxins, aflatoxins is actually the most predominant toxins, which occurs majorly in agricultural products or agricultural crops such as maize, corn, barley, millets, peanuts, etc. And the major fungus responsible for causing this kind of toxins or this kind of diseases are actually Aspergillus flavus and Aspergillus parasiticus. Now, if we look at the options provided, the option number C, that is Aspergillus flavus, is the correct answer because Aspergillus flavus is the fungi which produces aflatoxin. So, the correct answer for this question is answer C. Now, moving to the next question, under standard conditions in animal feeding studies, the weight gained in grams per gram of protein consumed by animal studies by an animal is termed as. Now, if we look at this question and the options provided, we can see that this question is actually from the topic of nutrition in the food chemistry section, and the question is asking about the method of protein quality evaluation, which tells about the formula that the weight gain per gram of protein consumed by an animal is judged by which of the given protein quality determination technique. Now, if we look one by one of the different options, we can try to decode what actually the answer is. So, the first is net protein ratio or NPR. So, NPR, it is actually the ratio of weight gain of the TPG, which is total protein group, minus the weight loss of the NPG, which is the non-protein diet group, divided by the weight of the protein consumed. Now, this is not the correct option because here there is a weight loss also involved in the formula. However, in the question, it has been asked that the formula should have weight gain per gram of weight consumed. So, moving to the next option, net protein utilization or NPU. So, the formula of NPU is retained nitrogen per intake of nitrogen into 100%. So again, this is not at all the correct option for this question. Moving to the next option, which is coefficient of protein digestibility or digestibility coefficient. It is the ratio of nitrogen absorbed to the nitrogen intake into 100%. And again, this is not the correct option because it does not involve weight gain per gram of weight of protein consumed. So moving to the fourth part, option D, which is protein efficiency ratio or PER. So the formula of protein efficiency ratio is the weight gain in gram per protein intake in grams. So this is the correct answer for the given question. So answer D, that is protein efficiency ratio, is the correct answer for this question. Now moving to the next question, xerophthalmia is caused due to the deficiency of. Now by reading this question, it is very clear that this question is from the topic of nutrition and we have to identify the deficiency which has been caused by the nutrient. So if we go through the deficiency of the particular options which have been provided, we can decode the answer. So the deficiency of thiamine or vitamin B1 is beriberi. The deficiency of pantothenic acid or vitamin B5 is actually very rare and it does not have any specific name. while the vitamin A deficiency is actually xerophthalmia and night blindness. So option C is actually the correct answer. However, also the vitamin C deficiency or ascorbic acid deficiency is scurvy. So as per this information, the answer C, that is vitamin A, is the deficiency of vitamin A caused xerophthalmia. Now moving to the next question, which one of the following statements regarding the hazard analysis critical control point or HACCP plan is not correct? So we have to identify the incorrect statement. So if we go one by the options provided, we can try to answer the question. So as per the statement 1 or the option 1, HACCP is a management tool for ensuring food safety. Now for this statement, we know the definition of HACCP, that it is a systematic approach for the identification, evaluation and control of food safety of hazard in food production facilities. That means it is actually a management tool which ensures food safety. So option A or statement A is correct. Now moving to the next statement or next option, HACCP involves 5 preliminary steps and 7 principles. So if we try to recall the principles and steps of HACCP, it actually involves 5 preliminary steps and 7 principles. So again, this option is also correct. Now moving to the next statement, HACCP is not effective without prior implementation of prerequisite programs or PRP. So this statement is again true, because without PRPs, there would not be a correct or effective implementation of HACCP. So this statement is also correct. Now moving to the next option, or the statement 4 or option D, HACCP plan involves establishment of corrective actions as second principle. So if we try to look at the second principle of HACCP, it is the determination of CCP, and the evaluation or the establishment of corrective action is actually principle 5. So this statement is the incorrect or the wrong statement. Hence, answer D is the correct option for this question. Now moving to the next question, the product of cabbage fermentation by leuconostoc mesenteroids is. Now it is very clear from the question that this question is from the food microbiology section of food technology section, and it is related from the topic of fermented foods. Now there are different type of fermented foods which are given, so we will try to look into what each of the products are from which category. So the first product is tempeh. So tempeh is actually an Indonesian food which is made by using fermented soya beans. So soya beans are being fermented to make tempeh by using the microorganism Rhizopus oryzae. Then natto and miso both are Japanese food products, and both are made using soya bean. However, the only difference between these two food products are that in natto, the fermented microorganism is Bacillus subtilis of the variety natto, while in case of miso, the microorganism is Aspergillus oryzae. Hence, we are left with the fermented food, which is sauerkraut, which is actually the fermented food which is being made by using cabbage, and is fermenting uses variety of lactobacillus lactic acid bacteria along with leuconostoc. So hence, this for this question, the correct answer is the answer C, that is sauerkraut. Now moving to the next question, which one of the following absorbents is not used as an ethylene absorber in active packaging of fruits and vegetables? So it is clear from this question that this question is from the food packaging topic of the food technology section. And if we try to look at all the given options, we know that among the given option, the option C, that is calcium hydroxide, is generally used as a carbon dioxide absorber and not as an ethylene absorber. So, apart from calcium hydroxide, all the other three options are used as ethylene absorbers because these materials help to delay the ripening and thereby extending the shelf life of fruits and vegetables because they absorb the ethylene, which is actually a natural hormone of plant which is responsible for the ripening of the fruits and vegetables. Hence, the answer C, that is calcium hydroxide, which is not an ethylene absorber, is the correct answer for this question. Now, moving to the next question, thermal resistance constant or Z value is defined as the change in temperature required to reduce the decimal reduction time of a microorganism by dash percent. So, this question is it belongs to the topic of thermal properties in the food engineering section. Now, if we try to recall the definition of Z value or thermal resistance constant, Z value is defined as the temperature increment or the increase in temperature which is needed for a 10-fold acceleration of the rate of the thermal destruction. That is the shortening of D value by a factor of 10. So, if the Z value is reducing, it means the D value is also going to decrease by 1 log or 10-fold. And a 1 log reduction is actually equivalent to 90%, or in short, 10-fold or 1 log cycle is actually equal to 90%. Hence, the answer is 90%. So, the answer for this question is 9090. Now, moving to the next question, processing of fluid milk at 72 degrees Celsius for 15 seconds is termed as. Now, this question belongs to the milk processing topic of food processing. Now, if we try to look at and try to explain all the given options, the HTST, which is high temperature short time pasteurization, it involves heating milk at 72 degrees Celsius for 15 seconds to kill the harmful bacteria while preserving the quality of the milk. In low temperature long time, which is LT, the milk is heated for 30 minutes, which is a very long time, at 63 degrees Celsius. While for ultra high temperature, the temperature is very high, from 130 degrees Celsius or even higher, and the time is 2 to 5 seconds. While the last option which has been provided, homogenization, is actually not a pasteurization technique but a mechanical process which breaks down the fat molecules and evenly distribute it into uniform size, which prevent any sort of cream separation. Hence, the obvious answer for this question is option A, HTST. Now, moving to the next question, which of the following fatty acid is or are known to increase the low density lipoprotein LDL cholesterol? Now, it is obvious that this question belongs from the topic of lipids. Now, again, we have to notice this thing that this question is actually a MSQ type question, that is multiple select type question, in which more than one option could be correct, and we can identify this thing by looking at the question that it is telling which of the following fatty acid is /R. So it is simply stating that more than one option can be correct. So we have to be very crucial while doing these types of questions. So now looking at the options one by one. So omega-3 fatty acids and conjugated linoleic acids are actually the fatty acids which are found in animal-based oil. For example, omega-3 fatty acids are primarily found in fish oils, and they are known to have a neutral and slightly beneficial effect on the LDL cholesterol. However, they do not increase the LDL levels. Similarly, conjugated linoleic acid, which are primarily found in meat and dairy products, they also do not have any increasing effect on LDL cholesterol. While the other two options, that is trans fatty acid and saturated fatty acid, both of them are known to increase the LDL cholesterol levels, thereby causing a risk of cardiovascular diseases. While trans fats are found in processed food and fried foods, saturated fats are found in animal products like butter, cheese, meats, and even some tropical oils like palm oil. So it is clear now that among the provided options, the option B, which is trans fatty acid, and option D, which is saturated fatty acid, are actually the correct answer for this question. Now moving to the next question, which of the following enzymes is or are used for the production of high fructose corn syrup from cornstarch? Now again, this question belongs to the topic of enzymes. And again, if we look carefully, we can see that this is also a MSQ type question. Now, if we try to recall the process of production of high fructose corn syrup, we can recognize and we can remember that there are three major steps which involves enzymes in it. For example, the second step, which is liquefaction. In the liquefaction step, the starch is break down into dextrin by the use of alpha amylase enzyme. Then in the second step of saccharification, the dextrins are converted to glucose by using the enzyme glucoamylase. And in the later step of isomerization, the enzyme xylose isomerase is used to convert glucose to fructose. Hence, it is evident that from the given options, the option B, which is beta amylase, is not used in the process of production of high fructose corn syrup, because beta amylase is actually used to break down dextrin into maltose, and since we do not require maltose for the production of high fructose corn syrup, the option B is actually the wrong option. But the question is asking which of the following enzymes are used for the production of high fructose corn syrup. Hence, the answer A, answer C, and answer D are the correct options for this question. Now, moving to the next question, which of the following is or are typical characteristics of a fungal cell? Now again, this question is from the food microbiology section, and the topic is characteristics of microorganisms, which include yeast, mold, and fungus. And again, if we look carefully, this is again a MSQ type of question. So we have to identify the different characteristics of a fungal cells. So now if we go through by all the options one by one, we can try to decode the answer. So the first option is presence of histone proteins. Now, since we know that in eukaryotes, the DNA is associated with a basic protein, which is nothing or histone protein. So in eukaryotes, DNA along with histone protein is a main component of cell wall. And since fungus is a eukaryote, so the option A is correct. Now the option B, the presence of peptidoglycans in the cell wall. Now we know that in prokaryotes, peptidoglycan is a major component of cell wall. However, fungus is not a prokaryote, so this option or this statement is not true for fungal cell. Moving to the next option, which is option C, presence of chitin in the cell wall. So fungal cells are known to have chitin in them as a major component, and fungal cells are encased in cell wall of a chitin, wherein chitin is a very strong but also flexible nitrogen containing polysaccharide, which consists of N-acetyl glucosamine residues. Hence, this option is also true for the characteristic of fungal cell. Now moving to the option D, which says presence of pseudomorine in the cell wall. Now, apart from bacteria and fungus, there is another group of archaea, and in archaea group of organisms, there are only certain specific microorganisms such as Methanobacterium and some methogens, which are known to have cell walls containing pseudomorine. So again, since fungus do not belong to the archaea group, this option is not true for the characteristic of fungal cells. So we can say that option A, that is the presence of histone protein, which is a characteristic of eukaryote, and since fungus is a eukaryote, this option is correct. And the option C, that is the presence of chitin in the cell wall, because fungus is known to have chitin in their cell wall as a major component. So option A and option C are the correct option for this question. So that's all for this part of solving GATE 2024 question paper for food technology section. Stay tuned for the coming videos for the remaining questions. Please like and share this video if you like the content, and do not forget to subscribe to our channel. Thank you.